Hi, my name is Daniel Gomez, and this is a bit for the basic research presentation. So this is on the structural basis of host protein hijacking in human T-cell leukemia virus integration. This study was done at the University of Minnesota and St. Louis University, supported by the Howard Hughes Medical Institute, and they're designated as an NCI cancer center. So this paper was published in Nature Communications on June 19th, 2020. And this is the paper that I'll be going over today. The main point of this paper was that the retroviral integrase forms a higher order nucleoprotein assembly called the intasome to catalyze the integration reaction where host factors remain poorly understood. So here, this research group used cryo-EM or cryo-electron microscopy to visualize the HTLV-1 intasome at 3.7 angstrom resolution. So what does atomic resolution look like? Well, we think in angstroms and in Fourier space. So starting with the human hand, looking at that, we can see a space on it, the square at 10 centimeters, looking at the skin. Zooming in, we have 1 centimeter. We can start to see the lines formed. Zooming in even further, 1 millimeter, we can see this, this X space. And then at 100 microns, you get even further into that groove and you can see the details there. Looking at a white blood cell, we can see 100 microns. You can see the whole cell, 10 microns. And then zooming in, we can see just 1 micron at 6. And then DNA, at 100 nanometers, we can begin to see the structure of DNA. Going at 10 nanometers, we begin to see the helices of DNA. And then at 1 nanometer, we can see the atom. So 1 angstrom is equal to 0.1 nanometers. And conversely, 1 nanometer is equal to 10 angstroms. So again, in this paper, we have 3.7 angstroms. So it's even smaller than 1 nanometer. Some of the world records in cryo-EM at the lowest resolution. Here in America, we have 1.34 angstroms, and that was done by Hua Chu at Stanford SLAC. And then over in Europe, they were able to hold the record at 1.25 angstroms, and that was Holger Stark over at the Max Planck Institute. So the structure together with the functional analysis revealed that B56 gamma or B gamma subunit of an essential host ribozyme, PP2A, or protein phosphatase 2A, is repurposed and then is an integral part of the intasome to mediate HTLV-1 integration. So looking at this essential host enzyme, it's involved in tumor biology. So we can see it here in this pathway, it being involved. And so you have a PTEN, AKT, mTOR pathway where PP2A can either inhibit the activation of mTOR, or if mTOR is active, PP2A can exacerbate that activation, leading to cell growth, survival, angiogenesis, migration, and invasion. So this essential host enzyme was deposited into the PDB as 2-IAE, and this was solved by X-ray diffraction at 3.5 angstroms. So there you can see the structure of PP2A. But getting back to the intasome, here's the overall structure of the HTLV-1 intasome. So starting with A, we see the target DNA and the viral RNA, the viral DNA. So target DNA, viral DNA, and then these catalytic CCDs of integrase in pink touching the DNA, and the CTD in pink, as well as the inner integrase CCD. So some of the components of this intasome are these integrase, right? IN, integrase, inner and outer. So there's outer integrase with the NTDs, CCDs, as well as outer integrase CTDs. So those are the linkers, right? If there's an NTD and a CCD with integrase, that's a linker. And then you have the B56 gamma on the outside. So B56 gamma on both sides on the outside, on the outer components. So the viral DNA is in black, the target DNA is in orange. And then if we rotate this 90 degrees, looking under it, we can see the viral DNA and these outer integrase CTDs. So on the outside, we see the B56 gamma in red, and then the other B56 gamma on the other side in light purple. Looking at the top view of the target DNA, we can see that in orange and B, and then these NTDs in green, as well as the CTDs in pink, and then the outer NTD integrase CCD linker in yellow, and the outer integrase CCD NTD in cyan, and that's interfacing with the B56 gamma here in light purple, and then this B56 is interacting with this outer integrase CCD. So if we rotate that 90 degrees, now we can see the viral DNA and the target DNA interaction with the outer integrase CCD, as well as the NTD there as well. And then again, the B56 gamma is participating as well. E and F are local resolution in angstroms. This is a map showing the local resolutions. So most of it is in 3.4 in blue, 3.4 angstroms, and then a little bit in green, which is 6.1, and then some of the DNA, the outer part is in at 8.8 angstroms. But there's not much at that resolution. But then if you rotate that 180 degrees, you can see the viral DNA more clearly there. So getting to the results. So they determine the structure of the HTLV-1 intasome. They use cryo-EM and single particle analysis. So this assembled a stable complex, including the HTLV-IN and a fragment of the human B56 gamma spanning the residues of 11 to 380. This had branched DNA molecule containing a viral U5 long terminal repeat LTR sequence and a target DNA, and this mimicked a concerted strand transfer reaction. So the presence of B56 gamma is essential for stabilizing this HTLV-1 IN DNA complex formation, and it's consistent with reports of a strong stimulatory effect of delta retroviral concerted integration reactions under certain biochemical conditions. So the molecular mass of the HTLV-1 intasome, or the strand transfer complex, STC, was formed and estimated to be 345 kilodaltons by SEC coupled multi-angle light scattering, or SEC MALS, and then 305 kilodaltons by mass photometry analysis. And then through cryo-EM density maps, this showed the complex to have a total mass of 320 kilodaltons. And this included the N-ion tetramer bound to the strand transfer product, DNA, and 2 molecules of the B56 gamma. Looking at the ion tetramer in the HTLV-1 intasome, it has a twofold symmetry that shares this conserved intasome core, or CIC, with the intasome assemblies and other genera of retroviruses. And then the ion tetramer consists of 2 inner catalytic and 2 outer non-catalytic subunits. So the ion comprises this N-terminal domain, or NTD, which is bound over the viral DNA major groove, and an NTD-CCD linker that contacts the viral DNA at both ends. So this is the catalytic core domain, or CCD, that engages the viral target DNA junction in the minor groove. And then the CTD, or C-terminal domain, fits between the NTD and the CCD of the same molecule in a cis-like fashion. So both the NTD, CCD, and CTD linkers of the inner catalytic IN run across a synaptic interface, and they're arranged anti-parallel of each other, interacting with both 5' overhang of viral DNA non-transferred strand. So the CTD of the non-catalytic outer ions further contribute to the extensive viral DNA interaction. This actually bridges between 2 viral DNAs attached to opposite strands of the target DNA. So the target DNA shows a kink at each of the viral target DNA junctions about 6 base pairs apart. And this results in a total bending of about 80 degrees away from the intasome core. There's also a zigzag trajectory of the target DNA that offsets the helical axis in the direction of that's perpendicular to the overall bending. And the PP2A B56 gamma ion interaction. So there's 2 molecules of the delta retrovirus for a specific host cofactor B56 gamma that are bound symmetrically to the core of the HTLV-1 intasome that flanks the viral DNAs, and this is known to cradle the IN tetramer. So both inner and outer subunits of an IN dimer on each side of the intasome fit into a concave surface of the B56 gamma. The CCD and CTD of the outer non-catalytic IN are bound towards each other in a banana boat shaped B56 gamma monomer, while the inner domain linker between the CCD and the CTD take a U-shaped conformation, and that's an anchoring interaction in the central peptide binding cleft of B56 gamma. Now there is this motif between 211 and 216 positions that have this LQPIPE sequence, and this is from the CCD-CTD linker, and we'll see this in a bit. And it's been known to be critical for HTLV-1 IN to PP2A B56, so it docks to a highly conserved binding pocket known as the LXXIE, or sorry, LXXIXE. So it's essentially LIE between 3 different X's. And that's a short linear motif found in a number of host proteins regulated by a PP2A. So these residues take a sharp U-turn near a little bit downstream of 219 to 223, that's SL, S, and K. And these interact with charged amino acids on the B56 gamma surface, including arginine 197. So the B56 appears to stabilize the IN dimer, and it helps organize these CCD-CTD linkers and position CTDs for viral DNA interactions. So here you can see a close-up view that's centered on the CCD-CTD linker and the outer non-catalytic IN, and that's right here. So here's the motif, the LQPIPE, short linear motif of the CCD linker. And if we rotate this 180 degrees, we can see the opposite side of B56 gamma. So here's the opposite side of B56 gamma and the NTD-CCD, CCD-CTD viral DNA with some 5' overhang, and the inner catalytic IN-CCD, and also the outer IN-CCD. So we'll go ahead and follow this trajectory from Q212 to Q212 over here in figure C. And here you can see a network of hydrogen bonds and salt bridges that mediate the binding of the IN-CCD-CTD linker in a U-shaped conformation. So this is the intermolecular and intramolecular contacts, which are yellow and orange dotted lines. So here's the intramolecular contacts in orange, and then these ones in yellow are the intermolecular contacts. B56 gamma plays an important role for HTLV-1 infection. So here we have some of the functional analysis. Here's a schematic diagram of HTLV-1 and HIV infection assay system. So here in A, we have a fluorescent signal that's generally following the removal of an intron here, disrupting this M cherry. It gets spliced out, and you have these producer cells. And then you have subsequent reverse transcription and integration of the viral genome in infected cells. So here in B, we have the shRNA control, B56 alpha knockdown, and B56 gamma knockdown. And essentially all of these BCD are relative infectivity assays of the indicated retroviral vectors in cells. So here in B, we have the B56 alpha knockdown. We don't see much change from the control, a little bit here, about 15%. And then in HTLV-1 B56 gamma knockdown, we see a significant decrease of relative infectivity to about 20 to 40%. So this shows that B56 gamma is critical to infection. In C here, we have an overexpressing B56 construct compared to eGFP. That's enhanced GFP, which shows a stronger signal than GFP. And here you have the other B56 constructs that are comparing to that. So you can see these constructs are 197A mutation, significant reduction in infectivity, as well as this L194A mutation. And then in D, you have an overexpressing 4 copies of a repeat peptide sequence with that the LIEXXX motif. This one's actually LPTIHE, which was the wild type. And you see compared to the cells expressing 4 in the ALA, you see a significant drop of infectivity in this wild type. It's expressing that specific motif. And then again, the p-values are here, which were done by an unpaired student t-test. So that's some of the statistics involved. And this brings us to the hypothetical model of the HTLV-1 intasome containing the PP2A holozymes. So here it is. You have our target DNA, our viral RNA, or viral DNA, and that's interfaced with the HTLV-1 IN integrase, as well as the PP2A B56 complex, and you have PP2A-C, or catalytic, and then the PP2AA for scaffold. So here's the HTLV-1 intasome, which contains the PP2A holozymes. Some of the methods in this paper were protein purification, HTLV-1 intasome preparation, cryo-EM imaging and data processing, model building and refinement, size exclusion and light scattering, plasmids and cloning for cell-based assays, cell lines, culture conditions, HIV-1 and HTLV-1 infectivity assays, mass photometry, in vitro integration assay, as well as the HTLV-1 integration site sequencing. And lastly, you have the protein binding analysis. They provided a table of the cryo-EM data collection and refinement and validation statistics. So they deposited their structure into the EMDB 21301, and that's its structure there provided. Bringing you to some of these metrics for cryo-EM data collection and processing. The magnification was at 96,000x, and the voltage in kV was 300. So this is done with the Titan Krios, seen in this image here. And we also have the pixel size in angstroms to be 0.8933, and the symmetry imposed is C2 symmetry. And then also important is this map resolution. Again, it was 3.7 angstroms. And then this included the FSC threshold at 0.143. Now, this is an important cutoff threshold that was proposed to make it comparable in resolution to X-ray crystallography. In the refinement, they kept the threshold. And then B factors, which is important for cryo-EM structural studies, they have protein, DNA, and ligand with these values. Lastly, I want to draw your attention to the Ramchandran plot. 96.29% was favored, and then 3.71 was allowed, and 0% were disallowed. This is the PDB deposit, 6VOY, and here you can see the structure, the target DNA, the viral DNA, and the enzymes. So to prepare for cryo-EM, you have to vitrify your sample, essentially freeze it through this nice VitraBot that was done in this experiment. And then here you have this Falcon 4 DED, or direct electron detector, which is some of the latest innovations in cryo-EM. Now I want to look at this EMDB deposit that I showed you on the last slide that was in blue. So this is 21301, and this is the 3D surface of the primary map view. So here you see the x-axis, and you can see the surface with the DNA, viral and target, as well as the enzymes. And then here's the y-axis, so you can kind of see it on its side, DNA and enzymes, the surface of the primary map. And then looking down, we see the x-axis, or looking up, you can see both DNA and protein. Here we have the primary map and fitted model of the PDB deposit. So here we have the X, Y, and Z axis. So here you see these alpha helices of the protein, and then the DNA strands in blue. So this map was fitted to the PDB model. And then you see the y-axis, so the strands are on their side, and then you see some of these alpha helices, and then some of these beta plated sheets inside, and then the z-axis is last. So this fitted model can be colored to include a score of atom inclusion residue, and this is viewed in different axes. So here you have the view along the x-axis. Everything looks good, looks the same. Y-axis looks just like the one before. Alpha helices and strands and some beta plated sheets. And then you have the z-axis. So there's the structure from the EMDB. So this is a unique solution by Delta retrovirus, and it hijacks and repurposes nuclear localized subunits of host enzyme PP2A to stabilize the intasome assembly. PP2A may direct the HTLV-1 integration into transcriptionally active regions via the interaction with certain chromatin associated proteins. So HTLV-1 TAX, or transactivator protein, has been reported to inhibit PP2A, the catalytic activity of that enzyme, and manipulate the cellular signaling and achieving activation of NF-kappa B, which is known to induce inflammation or neuroinflammation. So HTLV-1 intasome structure, this structure provides a mechanistic insight into the critical host pathogen interaction of an important human pathogen. And according to the, this framework allows for future development of novel therapeutic interventions against HTLV-1 infection and replication. Some future directions proposed by the paper were that, look at the phosphatase activity of PP2A. So investigate whether the B56IN interaction has, any outcome that involves the PP2A phosphatase activity. And thank you for your time. This is the HTLV-1 intasome, target DNA and viral DNA being integrated with PP2A and B56 gamma interactions. Thank you for your time, and I'm happy to take any questions.